In the last few sections of this unit, we're going to talk about enzymes. Enzymes are biological molecules that speed up chemical reactions. So you may remember from an earlier unit, anything that speeds up a chemical reaction is called a catalyst. Since enzymes are biological molecules that speed up reactions, you can think of enzymes as a biological catalyst. Enzymes are usually proteins, or we very often think of them as proteins. They don't have to be. There are actually many biological catalysts out there that are not protein molecules, but many times people think of enzymes as being proteins, and the examples that we talk about here will be proteins. The three-dimensional shape of a protein enzyme controls basically it controls whether the enzyme can attach to whatever it's supposed to attach to catalyze a chemical reaction. So if you have a protein enzyme that doesn't fold up in 3 dimensions properly, then it won't be able to attach to whatever chemicals it's supposed to attach to in order to speed up a chemical reaction, and it won't perform the chemical reaction. So these things have to fold up in 3 dimensions in the same way that the other protein molecules that I was showing earlier have to fold up in 3 dimensions. So here's sort of a cartoon version of a protein enzyme. That's this big gray blobby thing near the bottom of the screen. That's our enzyme. This material here is some chemical, and it's colored sort of blue and green, that can attach to the enzyme. So the enzyme has a 3-dimensional shape, and it has a little divot in its 3-dimensional shape, and that divot sort of snugly fits this chemical over here, and once this chemical sort of latches onto this divot, then the enzyme sort of can facilitate the breaking of whatever this chemical is into 2 separate materials, a green one and a blue one. So you can see this is a very generic example, but our enzyme is taking whatever this chemical is, attaching to it, snapping it in 2, and then the 2 products go on their merry way somewhere inside of the cell to do something else. And in this case, the enzyme's job is to just make sure that this this chemical is converted into these 2 products relatively quickly. And so there are a couple of terms that I want to point out. This is the enzyme. The little divot here, the part of the enzyme that is shaped so that it can sort of snugly fit this material, is called the active site. That just means the part of the enzyme that actually attaches to the molecule that is converted into something else. That's called the active site. For the most part in this course, when we have described a chemical reaction, let's say that this is chemical A getting turned into chemicals B and C, we've usually called the stuff that you start with, we've usually called that the reactant or the reactants, and these things are called the products. When they attach to an enzyme, sometimes biologists and chemists call them substrates. So this is just another name for a reactant. In this case, it's a reactant that attaches to an enzyme. So these are the other terms that I want you to remember. I want you to sort of have a general sense of what an enzyme is, what an active site is. That's this little divot that snugly fits the reactant. The reactant is very often called a substrate, and then the substrate is turned into products by the enzyme. As I mentioned in a previous section, I sort of said that proteins are like prima donnas. They, unless they are in the proper environmental conditions, unless they have the proper temperature, pH, and all of those environmental conditions, they will very often fall apart. They'll unfold in 3 dimensions. This is actually very easy to see indirectly using enzymes. So, as an example, there's a graph here. And on the x-axis of the graph, over on the bottom here, whoever did this experiment was changing the temperature. So they performed an experiment at 10 degrees Celsius, at 20, and at various different temperatures. On the y-axis, what they are showing you is they're testing one particular enzyme, and whatever reaction this enzyme catalyzes, apparently you can measure it. And if you're very high up on the y-axis, the enzyme is working well. It's catalyzing the reaction very well. It's converting reactants into products very quickly. If you're low on the y-axis, that means the enzyme is not working very well. It's not doing a very good job converting reactants into products. And then basically they said, look, here is one particular enzyme. How well does it work at different temperatures? And again, usually I ask these kinds of questions to the on-ground class, so you can pause at various points. But you can look at this and say, look, how well is the enzyme working at 10 degrees Celsius? Is it working well or is it working poorly? You can pause and think about that. And it's working poorly because the line showing how active the enzyme is very low on the y-axis. So the enzyme isn't working well. And then I say, is it working better at 20 degrees Celsius than it was at 10 degrees Celsius? You can pause and think about that. And the answer is that yes, it is working better because the blue line here is up higher than it was at 10 degrees Celsius. So the enzyme is working better at 20 degrees Celsius. And then I say, well, what temperature does the enzyme work best at? You can pause and think about that. And people usually say it works best at a temperature that's a little bit less than 40 degrees Celsius. So in the high 30s degrees Celsius it works best at. And then you can see that if you deviate too much from this temperature, from the high 30s, if you go too far and make the temperature too hot, the protein stops working. And it's not working, this enzyme isn't working because it's unfolding. This protein is denaturing. The enzyme doesn't assume its normal three-dimensional shape. It can't attach to the reactant and convert it into product anymore over here, and so it works horribly. Same is basically true at the very low temperature. And the enzyme works best in the high 30s. In reality, this enzyme seems to work best at about 37 degrees Celsius. For those of you who know what's special about 37 degrees Celsius, for those of you who don't know, 37 degrees Celsius is body temperature, it's human body temperature. So whatever this enzyme is, it probably either came from a human or from an animal that has a similar body temperature. And so you can imagine that the enzymes that work in our body, they are accustomed to working best at our body temperature, at 37 degrees Celsius. So, and if you deviate too much from that temperature, if you try to get our enzymes to work at 50 degrees Celsius or at 10 degrees Celsius, they work horribly at that temperature, in large part because they are not assuming the standard three-dimensional shape that they're normally accustomed to working in. And if they don't assume that three-dimensional shape, then they're not going to do their job well. So this is just an example of where you can see how a protein or an enzyme, in this case a protein enzyme, will only do its job if it's working under the appropriate temperatures. And so this particular temperature, 37 degrees Celsius, is sometimes called an optimum temperature or a temperature optimum. So that's just an example of how certain types of proteins will only work under certain environmental conditions. This picture here is a picture of a hot spring at Yellowstone National Park. If you have never gone there and ever have the chance, I would recommend it. I've been there once. It's absolutely beautiful and smells like northern New Jersey. The reason it smells like northern New Jersey is because Yellowstone National Park is basically on top of a gigantic volcano. And because it's under a large volcano, a lot of the lakes in the area are heated up to near boiling temperature. So this is a lake, and this is steam coming off of the lake. So it's incredibly hot water here. And these colored stripes that you see, this sort of green stripe, this yellow stripe, this reddish-orange stripe, are actually bacteria that have evolved to grow, and their optimal temperature is they grow best at very hot temperatures, at near boiling temperatures that are near the temperature of boiling water. And basically, if you could extract some of the enzymes from these bacteria that are accustomed to growing at very high temperatures, you'll see that the temperature that the enzymes from these bacteria, the temperature that they work best at is near boiling temperature. And if you actually try to get those enzymes to function at our body temperature, they function horribly at our body temperature. Basically for the same reason that our enzymes function horribly at 10 degrees Celsius or at 50 degrees Celsius. The enzymes in the bacteria here are just not accustomed. They haven't evolved to function properly at what they consider to be strange temperatures. They're more accustomed to functioning properly at near temperatures that are near boiling water temperatures. So this is just another example of a situation where enzymes will only work at the temperatures that they're sort of accustomed to working at. You can also see this type of effect with pH and enzymes. So the idea is that many enzymes have an optimal pH that they work best at, and if you deviate too much from that optimal pH, you will unfold the enzyme or the protein and basically cause it to be unable to do its job. Here, this is on the x-axis, we are altering the pH, so pH 7 is neutral. In this direction, it's acidic. In the direction toward the right, it's basic. And then we're looking at some particular enzyme. Don't know what it is. But if you're low on the y-axis, the enzyme is not functioning very well. And if you're high on the y-axis, the enzyme is functioning well. And again, I can ask what is the pH that this particular enzyme works best at? And you can pause and try to figure that out. And in this case, the pH that this enzyme works best at is slightly above 7. So pH just a little bit, just a hair over 7. But if you deviate too much, if you make the surrounding conditions too basic, or if you make them too acidic, the enzyme doesn't fold properly and it doesn't catalyze its chemical reaction. Sometimes I will ask, where do you think this enzyme is found? You can pause and think about that if you'd like. My suspicion, and I probably got this from your book, so there may be details elsewhere in your book, my suspicion is that this enzyme is found in the blood because the standard pH of human blood is slightly above 7, and that's the optimal pH here. So that would be my guess as to where this enzyme is normally located. But again, this is just another example of enzymes needing very specific environmental conditions to function properly. This is another table taken from your book, and it's just sort of illustrating the point again with different enzymes. There is an enzyme called pepsin. It is found in your stomach, and as you know, your stomach is relatively acidic. And if you look at the optimal pH that pepsin works at, pepsin's optimal pH is somewhere between 1.5 and 2. So pepsin works best under acidic conditions because that's where it's normally found. That's where it's normally supposed to be doing its job. And many of the other enzymes in this table work best at pHs that are closer to neutral pH. pH 6.7 to 7 and pH 7. That's not always true, but for the most part, enzymes will have an optimal pH that reflect the environment that they're normally found in.